Okay, yeah, welcome, Zihao. Is that how?
Yeah, Zihao. Yeah.
Yeah. It's a great pleasure to see you today. And I think for our seminar series, we also want people to know about the people who made the discovery and the story behind it. So we
Mm.
want to have this mini interview so people know better about you. So how about you tell us a bit about your background, as people are still tuning in, and people can also know about you a little bit more.
Yeah. I get, you mentioned this to me before, so I added one more slide.
Great. So you are trained as a physicist, no?
Yeah. Yeah. So like, maybe we can go this.
Yeah,
So,
yeah.
yeah, this is basically my academic background. So, I got my Bachelor of Science in Physics in 2015 from the University of Science and Technology of China, in Hefei. And during that time, my interest was using computer simulation to understand the dynamic behavior of what we call active systems. It can be, it's kind of a model system. It can go from bacteria to live animals like birds or many fishes. When they move together, they form certain kind of pattern. That's what we call active materials. That's my undergraduate research. And during that time, I also spent a few months at University of Oxford. That time, I'm studying a more advanced theory using what we call phase field model to model this active system. So my training in the undergrad is all in the theoretical physics as perspective. And after I finished my Bachelor of Science degree in physics, I started to feel like um, I would like to know a little bit more about how real world actually actually function. So I decided to move towards um, experimental science. So I joined the material science and engineering department of material science, yeah, of the University of Illinois at Urbana-Champaign. So that time I got access to a very interesting technique called liquid phase transmission electron microscope. So these technique, what they can do is they sandwich a small droplet containing nanoscale objects, and you can put this liquid droplet into a high vacuum chamber, and using electron beam, these allow you to visualize the nanoscale dynamics inside the liquid environment. So that was my PhD research. So as you can see, there's a connection, but it's completely different field. And then well, after I'm done in finishing my PhD in 2020, I feel like material science, we are working on these nanoparticles, but the impact is a little bit limited at this moment because I think many aspects is that we are lacking of control, we lack of understanding. I want to learn more. How can I increase the impact of my research or my vision of academia research? Then I decided, I told my advisor I want to maybe learn some biology, how to design imaging system for biological system. That's why in 2020 to 2024, I joined Stanford as a postdoc fellow, and that at my main advisor is still in the material science department, but we are developing new imaging methods for live animal research. So that's kind of the topic that I'm going to talk about today.
So is the Holmes group always working on this live tissue, live animal imaging?
Mm,
How do you find this collaboration or like a co-mentorship?
yeah, yeah. We can talk about this first. So at the beginning when I joined the Holmes lab, the Holmes our my postdoc advisor, their vision is using nanomaterials for live animal neuromodulation. It's totally different. So so then they have a one question that is bothering them is when you use electromagnetic field to stimulate nanoparticles inside live animals, the interaction of the electromagnetic wave with the nanoparticles is not very clear. Because I have a background in physics and material science, so my advisor got me there because they want to understand how this electromagnetic wave interact with nanoparticles inside live animal environment. And I started to dig into theory, look at literatures, design experiments. I've done all these things for the first year of my postdoc, and the project failed. So, and we designed some fundamental experiments try to measure the electromagnetic wave interaction with the nanoparticles, and it turns out to be very, very challenging. In the because when the environment is complicated, reading accurate light signals from the instrument is tricky. I couldn't get consistent experimental results. So probably at the summer of 2021, we started to think, what else can we do? And we realized that some of our experimental data suggested that the scattering of tissue actually uh, changed when we introduced absorbing molecules. So that's kind of the starting point of what I'm presenting. So um, it's actually I think a lot of discoveries are like this that we never, this is not the direction we planned for, but in the end, we find something interesting that is during our initial design, and then that leads to something more exciting to all of us.
I guess I will let you present. I do have a
Mhm.
lot of more questions we can follow up. I think it's better that you lay the foundation first, and I think people cannot wait to hear your exciting findings
Yeah,
and
yeah,
how you put them into applications that your lab is trying to do. And yeah,
right.
we'll hear you first, and then I will follow up with the interview, pick up from here.
Yeah, yeah, yeah. Thank you so much. So let me still go back a little. So let me just my name is Zihao, and I'm currently an assistant professor in the University of Texas at Dallas. So I'm in the physics department, but my lab is specifically belonging to a biomedical science program. So inside this program, we hope the understanding of physics, chemistry, and even mathematics can help resolving some of the biological challenges. So that's why today we are going to talk how we are going to use absorbing molecules to make tissue transparent. So as you can see, here's a photo that we took. We are extracting these molecules from snacks. So this is just a photo to illustrate how easy you can access these kind of molecules. And later, you're going to see this tube again at the end of this talk. So uh, yeah, so I will briefly talk about this. This is my academic career. I started as a Bachelor of Science in Physics and then did my PhD in Material Science. And I started to get developing this novel imaging system for live animals when I was doing postdoc at Stanford University. So generally, my group is very interested in how to understand dynamic processes, and this is why you can see all the connections in my past research experiences, either from computer simulation or electron microscope or optical microscope. So understanding this dynamic process is extremely exciting to me because for us, we feel like being able to move around is important for us. For example, we can look for food, look for shelter when we feel danger. And these dynamic motions are all realized by the complicated movement of individual cells. And cells can divide, cells can migrate, and this complicated motion of cells are realized by the even more complicated subcellular motion movements. And to visualize these movements, you can imagine these are realized by proteins. Uh, they are to control the molecular behaviors. So if you look at all these dynamic processes, they actually span different length scales. So as you can see, our human, we are on the meters and our organs are pretty big. But if you want to understand individual cells, we need to look at cells at tens to hundreds of micron. If you even want to study these nanoscale machineries, we want very high resolution that is go to 1 to 10 nanometers. So, motivated by techniques to visualizing these dynamic processes, so I started to look at this imaging range as well as imaging depth, imaging modality landscapes. So if we want to study the dynamic processes um, inside our body, the most common technique we'll probably think about is probably MRI or ultrasound. These are all non-invasive tools that can go very deeply into our body, but they are relatively limited by the imaging resolution. So here is an example of what we call super resolution ultrasound imaging. Super resolution ultrasound, it can go to down to about 100 micron, but this resolution is still, you can see it's much larger than the individual cells. So it is challenging to using ultrasound to visualize cells at this moment. So for most of the cellular studies, we probably would go to optical microscopy. And if you want even higher resolution, you could even go to super resolution microscopy. And here we are showing a rotating fluorescent beads driven by a DNA origami. And using super resolution microscopy, we can actually track this single molecules down to very high accuracy. But you may have noticed that for super resolution microscopy, you need fluorescent labels. And what we are tracking is actually this location of the single fluorophore instead of the intrinsic morphology of this molecule. So if we want to indeed look at the intrinsic morphology of the nanoscale features, we'll probably go to electron microscopy. And here are triangular nanoparticles that I made before and the electron microscope image I took. But as you can see now, it's just a static image. It is very challenging to conduct dynamic imaging experiments in electron microscopy environment. So let's take a look at this imaging modality landscape again. So as you can see, when we go from um, deeper the techniques that can go very deep to techniques that we can have very high resolution, we can see that it actually following this direction, which means that um, when we have higher and higher resolution, it seems the penetration depths are getting shallower and shallower. So why do it seems that this is actually limiting a lot of our biomedical research capabilities because of this change of imaging resolution as well as imaging depth. So can we make some improvements to fill in the gaps inside this imaging modality landscape? So what I'm going to introduce the work that we recently published is that we try to fill in the gap to make us visualizing deeper features using optical microscopy, where we can change the tissue transparency of a live animal. So in my PhD, I've been working on with electron microscopy so that we can actually use electron microscopy to study dynamic behaviors. But for today's talk, we are just going to focusing on our recent work where we make live animal transparent to make to use optical microscopy to visualize deeper features. So let's start with a relatively simple fluorescent imaging setup for live animals. So as we can see, here we have an excitation laser, and then we shine onto the animal, and then we collect the fluorescence signals going through the emission wheel, and then the signal are collected by the camera. If we take a closer look at this region where light interacts with the animals, we can see that external light need to shine on the surface of the tissue and we get reflected and it will get scattered and absorbed along the pathway until it can achieve the fluorophores. And similar processes also happens for the fluorescent signal that is going to get scattered and absorbed until it can be going out of the animal to be collected by the detector. So let's take a closer look at this scattering and absorption processes. What does it mean, scattering? So it means that when light is propagating inside the tissue, it changes its propagation direction over time. So as here, a simple illustration is that light, when a scatter event happens, this light changes its propagation direction. To quantify these processes, we can introduce a mean-free path so that this mean-free passing is inversely related to a scattering coefficient, as illustrated here. And similarly, we can introduce an absorption coefficient, which describes how far the light can go inside the tissue until it's getting absorbed. So then we have these two fundamental processes inside our tissue. So generally speaking, which one is more dominant that is limiting the capability of optical imaging? So a lot of times when I ask these questions, people would think about absorption is more important. It is true for absorption to dominate in certain cases, but most of the tissues actually is different. So there's this very simple example. If you shine a laser onto your finger, you will probably see the laser can illuminate your whole finger. So then let's take a look at these two extreme cases. One is the case when the scattering is much more dominant than the absorption. What you will see is light getting scattered into like a bulb, much larger volume. While when the absorption dominant, we see that light actually don't changes direction too much, but getting terminated inside the tissue. So as we can see from the simple experiment I mentioned before, is that when you shine a laser pointer, you will see that it's actually more close to this case. That's what we want to illustrate. The first point is that in most biological tissues, scattering is actually more dominant than absorption. And using scattering coefficient and absorption coefficient as a quantitative measure, you can see that the scattering coefficient is about 10 to 100 times bigger than the absorption coefficient in most of the visible to the near infrared wavelengths. So scattering is more dominant, that we see, but why scattering? Why the biological tissue has such a strong scattering? So um, when you think about this, if you don't understand the fundamental physics behind it, this maybe sounds really challenging how to think about how to model this. But the we can give you a very relatively simple example as bubbled water. So you've probably seen this in a lot of real times when you have soda, when you have air gas inside the water, you will see the whole system become opaque. So let's just think about what is inside the system. We have water, which is transparent. We also have gas, which is also transparent. However, when you mix water and gas together, they become non-transparent. So let me just emphasize that it is not because water or gas is not transparent. It is only when you mix these two together that this becomes not transparent. So why could that happen? So it is very simple. Probably everyone knows that when light goes through air-water interface, it will changes its propagation direction. So inside this air-water bubbles water, when light trying to go through the system, it will face numerous interfaces, and every time it changes direction. That's why when light cannot go through this bubbled water very easily, and it turns whitish, which means it's not transparent anymore. So biological system is actually no more complicated than this. So it's just like inside biological systems, we have a lot of optical interfaces. We have lipids, we have fibrils. All these have a different optical properties, and light will change its propagation direction every time it faces these interfaces. So a more realistic schematic is more like this. We have a schematic of cell and it's embedded in a complicated biological environment. And inside this environment, water has a relatively low refractive index, about 1.33, while you have relatively high, you have subcellular components which has a relatively high refractive index, and some of the functional molecules they can have a really very high refractive index, such as fibrils. So let's imagine we draw a line across these cells, and we will see that it starts from relatively low refractive index, but every time it faces this interface, it will change its refractive index. And uh, we follow this path length, and we will see that this value varies a lot. So it is exactly the same as what we referred before as the bubble water, that light will get scattered into all different directions. And this is the reason why our tissue is not transparent to light. And an easy way to make tissue transparent is there a way that we can reduce this refractive index profile? We make it relatively close in terms of all the optical components so that light can go through the tissue without scattering. So this is seems like yes, for this audience, everyone probably heard of tissue clearing. So this is a fundamental principle for all the tissue clearing technologies. So you probably know that in protocols such as Disco, what we do is we exchange the external aqueous environment to a high refracting index fluid so that light can go through this tissue without scattering because everything has a relatively high refracting index. On the other side, you can remove all high refracting index components and only leave the low refracting index component, and then light can also go through without scattering. So these two methods can both achieve optical transparency, but they rely on the similar concept of reducing the refractive index differences but one is making everything high, but the other is making everything low. So these are previous work that has got a lot of attention, but from a physics perspective, we started to ask one more step. So why different components have different refractive index values? So to explain this, we need to go to a little bit of math and physics. So what is refractive index? So let's revisit the physical meaning of refractive index. So refractive index is a physical property that describes how light propagates inside one material. So as we can imagine, we can have electromagnetic wave or light um, of different wavelengths or different colors, and they propagate inside a material with uh, permittivity value epsilon. A very easy model. And then I won't go to details about Maxwell equations, but if you use Maxwell equations to solve the electromagnetic wave inside this medium, what you will get is a plane wave solution that is showing here. And this plane wave solution, inside this plane wave solution, we have this wave vector k, and wave vector k is the vacuum wave vector 2 pi over lambda times the refractive index, which is the square root of this permittivity. So what is very, very important here is that you will see refractive index has both real and imaginary part inside the values. So a lot of times, previously when we were talking about water has refractive index of 1.33, we are only talking about the real part of refractive index. We ignore the imaginary part of refractive index. So what is does it mean to have imaginary components inside the number? So let's since it may have been a while since you heard about complex numbers, let's do a little bit of revisit of math. So complex number means that a value has both real and imaginary part. So real part is there's a real value, while imaginary part has this real value plus times an imaginary unit, and this imaginary unit, the square of imaginary unit is -1. So um, the other way of expressing complex number is using the Euler's formula, where you can express it in this using a phase angle, and this exponential I phi can be is related to the cosine phi and the imaginary sine phi. And then with this mathematical information in mind, now let's take a look at this wave function again. So we have the electric field in the initial field magnitude times the propagation of the field. And now we know that k, this wave vector, is a complex number. And then if you plug this back into this propagation term, what you will get is we will have this electric field. One is relating to the propagation of the field, and the real part of the refractive index actually determines the phase speed. And we have another exponential decay, which basically physically tells what is absorption of the medium, how much light decays when it's propagating along this direction. So as we can see here, the important take-home message is that refractive index has both a real and imaginary part, and they correspond to two different physical meanings. And now we are going to talk about one of the questions that Charles shared with me earlier. What is the importance of Kramers-Kronig relation? So when one physical property has both real and imaginary components, these two are not independent. So you cannot change one without changing the other. And mathematically, these two properties are correlated by what we call Kramers-Kronig relation. So this mathematical expression is a little bit intimidating, but simply I can help you go through that. Is you can see the real part of refractive index is related to the imaginary part of refractive index in all the wavelengths. So if you know the imaginary part of refractive index, you can actually calculate the real part of refractive index without measurements. And other way of saying that is if you change the imaginary part of refractive index, the real part of refractive index is also going to be changed. So let me illustrate that is that if I introduce imaginary arbitrary peak in the imaginary refractive index, the real part of the refractive index is going to be changed following this peak location. So what is the imaginary, as we just talked about, what is the imaginary part of refractive index? This is corresponding to absorption. And the real part of refractive index is related to a lot of this scattering properties. So this tells me that it seems that I can utilize this strategy to actually change the real part of refractive index to reduce the scattering. So let me go through this what I just said very carefully again. So let's start from water's refractive index, real part of refractive index, which is approximately 1.33 over the visible to infrared wavelengths. And if I introducing an imaginary peak, so the imaginary refractive index peak into this uh, water, which is simply adding some absorbing molecules, the real part of the solution refractive index is going to be changed following this absorption peak. And in certain wavelengths range, we can see that real part of refractive index can actually reach to a level comparable to the high refractive index high refractive index components such as lipids and collagen. And what is even more important is that these regions actually has relatively low absorption, which means light can penetrate through without being absorbed. So it sounds like a really nice design, but uh, is this material really exist? So we want materials that actually can change the refractive index in a very efficient way, and we want it to have relatively low absorption as well. And importantly, if we want to do biomedical research, we want it to be safe for animals. So here is a numerical example, but let me show you how it looks in real materials. So here is a characterization of the imaginary refractive index and real part of refractive index using a tool called ellipsometer that is commonly used in the semiconductor industry. And we can, by measuring this separately, we can see that when we have this absorption peak, we can actually indeed observe the change of the real part of refractive index in the nearby wavelengths. And similarly, since we can control this level absorption uh, by the concentration, as you may know from the Beer's law, we can actually also control the refractive index of this solution using the concentration. Here, let me introduce two other parameters. That one is alpha, which describes how the absorption depends on concentration, and the other is beta, which describes how the refractive index depends on concentration. So using this alpha and beta, this gave me a method that I can actually compare different materials. I started to measure this for for about 20 different chemicals. And what is interesting I find is that it seems when the absorption peaks go to longer and longer wavelengths, the molecule is more and more efficient in terms of absorption and changing the refractive index. So at the beginning, I was very confused by these results, why this happens. And I presented these results to our collaborator at Stanford, Professor Mark von, and he told me that this can be simply explained by a simple Lorentzian model in the textbook. He told me to simulate dye molecules that are absorbed at different wavelengths using this equation. And surprisingly, you can see that when I simulate molecules absorbed at 100, 200, and 400 nanometers, we can see the absorption level of the molecules getting increased, and as well as the capability of changing the refractive index. So from this model, it seems suggesting that we should use molecules that absorb at relatively longer wavelengths so that they are going to be more efficient in terms of modulating the refractive index. However, as we mentioned before, we also want the materials to be transparent in certain wavelengths so that light can go through these wavelengths without being absorbed. So in the end, we add another selection criteria, which is the average absorption between 500 to 700 nanometer, and average refractive index modulation in 500 to 700 nanometer. So by putting this together, as we can imagine, we want material that in this wavelength with relatively low level of absorption, but relatively high level of refractive index modulation. And that's why this diform molecule stands out as one of our candidates. So the last question is this chemical actually going to be safe for biomedical research? So what is this diphor? This diphor molecule is actually widely used in food industry. It is used to add the yellow color into the snacks and drinks. So if you check in the future, if you're interested, you can when you buy a bag of Doritos or get a orange colored energy drink, Monster or others, you can check the ingredients. Usually when they add this yellow color, it's a combination of two industry dyes, is yellow 5 and yellow 6. So what we are reporting here is yellow number 5, which is named taurzine, and this has been widely used in food, so we think we are so lucky that this dye is potentially safe for animal research. So now we have find this potential ideal candidates for our application. So how does this really look in experiment? So before we go to biological systems, we first did this experiments by utilizing a silicon gel. So what we prepared is this is an agarose gel with doped silica particles inside. Because silica particles has a higher refractive index than the agarose background, um, this phantom, we call this a teacher phantom, and you can see this phantom is actually completely opaque, and light cannot go through this phantom because of the scattering between the water and the silica particles embedded. And when we start to introduce tartrazine molecules, we can see that this phantom obviously become yellow, which is the color of this dye, but it's also gradually becoming transparent. So I would like to pause here for a second that you can think of this system again. So we are introducing absorbing dye molecules into a strongly scattering medium, and this scattering medium become transparent. So if you don't understand the physics behind this, you would think like this is like a magical trick. How could that happen? But now you understand the physics is actually not very challenging. So we can also quantify this process, this clearing performance. As we can see at the beginning, the transmittance of this phantom is almost zero across all the wavelengths. But once we introduce these dye molecules, it become more and more transparent in certain wavelengths, and we can see that some part of the wavelengths get increased faster than the others. And we can also build a mathematical model to describe this, and we can see our measurements matches with our theory very well. So this is a mesoscopic demonstration. Apart from that, we can also utilize what we call a resolution target. So this is like you go to clinic to do a vision test, but this is a vision test for the optical imaging system. So what we do is we prepare patterns with a relatively large feature and relatively small feature. So the smallest feature here is about 1 micron, which is matching the size for subcellular imaging. So as we can see, when we don't introduce the dye molecules, the pattern is very blurred, and we cannot achieve higher accuracy imaging. But when we start to introducing these dye molecules, we can see that the pattern gradually become clear. And our collaborator Yiqiu in Professor Mark Bronxmark's lab, he also conducted numerical simulation to help us illustrate that light can indeed be refocused to achieve high resolution imaging. So, but if you pay close attention to one of the column, you'll probably see that when we start introducing dye molecules, it becomes clear, but at even higher concentrations, this becomes blurred again. So why this happens, we can actually use this resolution target as a quantitative measure of imaging contrast. So here we have a clear image and a blurred image. If we draw a line across this clear image, we will have high intensity, low intensity, while if you have a blurred image, this is almost constant. So we can define this MTF value, which is the maximum minus minimum over maximum plus minimum, and this will go to 1 for high resolution, high contrast image, goes to 0 for low contrast image. So we may expect this to only at the beginning we have the value close to zero for all these features, and when we started introducing dye molecules, it's getting increased. However, this is only for one wavelengths. In certain wavelengths, we can see that these first increase and gradually decrease again. So this is because we are talking about reducing the refractive index contrast. It is not just increasing the refractive index in water, it is actually matching the refractive index. So that's why it's not always the higher the concentration is better. We actually want to achieve an optimum imaging condition. So when we analyze these data by calculating what is the difference between refractive index between the particle and the background, we can actually overlay all the data together and we can see the optimum imaging condition is only achieved when we have when the refractive index difference between these two components are getting very closer to zero. So we have been talking about model system. So how about actual tissues? So this is some recent results our postdoc did my postdoc Waka did at UT Dallas. So as you can see, a chicken breast tissue, when we place it on this 1 millimeter grid, we cannot see through the patterns because this tissue is scattering as we described before. But after we soak it in this dye solution, we can make this chicken breast piece become completely transparent and clearly visualize the individual grid behind this pattern. And you can also show up hide other informations such as the logo of our school. Um, and what I want to emphasize is that um, this is images just captured by our camera or phone camera, which means that it's very close to what we are seeing. So since a lot of people are interested in how these experiments are done, so here's a video that maybe help you understand the experimental processes. So this is a chicken breast tissue um, that we haven't done anything with it. We just put it into a petri dish, and we cannot see the pattern very clearly. But once we put it into this dye molecule, you can see this piece of tissue gradually become transparent, and you can see the time is um, only about 10 minutes when it becomes transparent, and it's getting there's not much change after 30 minutes, right? But this time, how much time is required actually relates to the thicknesses. If the thickness is doubles, the time required going to be 4 times longer, right? So as you can see, this process is very, very simple. So since we are just trying to match the refractive index between the background and the cellular components. And because it's simple experimental procedure, and then we started to think maybe we can adapt this method to live animals to make part of the animal tissue transparent. So this time, now we're starting to talk about live animals. So if we shave the belly of the mouse, and this is what we see, we cannot see the internal organs very clearly because of the scattering that I introduced before, that our skin is blocking the light that we cannot visualize the internal organs. So when then we prepare the soft gel containing our dye molecules and apply to the surface of the mouse skin, and start doing some massage to help the dye diffusing into the skin. So we can see turn the skin into a transparent layer, and this allow us to visualize the internal organs. And I think a lot of you may know better than me. You can see the liver, small intestine, large intestine, as well as bladder, without opening the mouse body. And one more important thing is that this is actually similar to before. This is covered by um, color camera, means it's very similar to what we observed by our eye. We actually don't require any additional imaging, complicated imaging setup to capture images like this. So because this is our method is so easy to apply to live animals, this this is actually not just a single image. we can actually take a video and we can, on one side, we can actually rotate the animals and examine the internal organs from different angles. We can also keep it stable and visualize the peristalsis inside the live animals. So just for your references, um, this is how people used to do if they want to investigate the internal movement of the GI tract. They either need to open the mouse belly and take the GI tract out, or they replace a large piece of tissue with artificial materials so that that allows visualizing the internal organs or the movement of the GI tract. As you can see, there's a great advantage of our method in comparison to the previous techniques. So in my postdoc trainings, I am particularly interested in the movement of the GI tract, the gut. So as we know, gut is one of the largest organ in our body, and it controls the digestion, absorption, as well as excretion of substances inside our body. And all these functions are realized by the dynamic movement of the GI tract. So nature has evolved a very interesting way so that it can send stimulating and inhibiting signals upstream and downstream to achieve this alternating relaxation and contraction movement so that food can be propagated unidirectionally along the GI tract without causing mass inside our body. So during my postdoc training, my co-advisor, Julia Koshby, she's in the neurosurgery department, and she's an expert in studies of neurocircuitry that is controlling the movement of the GI tract. And through her mentorship, I got to know a lot of how to utilize genetic tools, genetic mouse models to understand the nervous system. So here, the mouse line I used is a chat creatine tomato mouse line. So a specific group of neurons in the enteric nervous system is labeled by the TB tomato fluorescence signals. So typically what people do is you cut this out and bring it to a confocal microscope, and you can examine the structure of this neural network. And now we have a way to make this transparent. And you can see what now how we can kind of change this experiment. Instead of taking the gut out, we can make part of the tissue transparent, and this allows us to directly visualize the neural networks without opening the mouse. And this is epifluorescence imaging. And similar to before, this is not just a static image. We can actually visualize the dynamic movement of this neural network inside the animal body without any surgery now. So let me skip a few slides. I think time is a little limited. So we can actually analyze the patterns of all different movements inside the animal body. And this may give new information of how the contraction, expansion, and rotation inside the animal, inside the gut. So apart from studying the GI tract, we can also apply this to the head and utilizing laser speckle imaging to visualize the blood flow patterns. And we can also apply this to the limb of the mice and utilize two photon microscopy to visualize the individual muscle band. So here I would like just to highlight that this individual muscle band is about 2.5 micrometer away, so that actually demonstrate our imaging method can actually be compatible with high resolution imaging. So since our publication, we actually got a lot of attention, and this is what I showed before at the beginning, that we have this tube. I extract some of the chemicals from the snacks and took a photo, and this got to the CNN. And then people were saying that we are making steps towards an invisible man. And what is more important to biomedical research, I think a lot of people realize now it seems like we could potentially have a transparent mouse model for biomedical research. And following that, I would like to show that the most recent work that is published on bioRxiv, how people utilize our technology for their own research topics. So in this recent work on bioRxiv, they first reproduced our experiments, and following that, they did something beyond us. They applied our technology on the back of the mice and tried to visualize the spine and spleen. So in another example, this is a group in Duke where they utilize our magic molecule tartrazine to visualize the porcine eyes using OCT imaging, optical coherence tomography. So I think the take-home message is that our method will change the optical transparency of the tissue, and we don't actually pose any requirement and on the post imaging techniques. So in other way of speaking, we can improve the performance of all the existing optical imaging modalities for live animals, just very similar to what has been demonstrated here for optical coherence tomography. So I would like to end my talk here. So this most of the part of the work is actually done at Stanford. I would like to acknowledge my group, my postdoc group, Guosong Hong, and my co-advisor, Julia Koshby, is my important collaborator in Proxima, as well as support from the Wucai Neuroscience Institute at Stanford. I was supported by them. I'm a postdoc fellow there, and um, our work involves live animals. We appreciate contributions from all the live animals. And this is our new group at UT Dallas. It's a beautiful new building. And this is my first graduate student, Matt. And we are actively developing new materials and hope physics can help us resolving more interesting biomedical challenges. Thank you.
Thank you very much, Zihao. And I just will wrap up with one or two more questions, and I will open it to the audience. So if
Yes.
any audience, just to make an announcement. So if the audience want to ask a question, how about you raise your hand or like somehow let us know, like unmute yourself, and like even better if you can show your video, and so that the speaker can see you. But let me just start with one question. This absorbing material, when you apply it, now you can adjust the RI and so can see better. I just wonder, is there any trade-off by adding this absorbing
Mm.
material? Now, are you losing the signal intensity? Do you need a stronger light, like power source, like to shine the light on it?
Yeah, wonderful question. This is one of the reviewers' comments. So, so it's a interesting. So maybe I can go back actually very quick, easily. I think. Uh, so this is one of the things when we start working with these materials, we are actually quite concerned. We don't know if we can achieve this or not, just because of the reason you mentioned. So what we want material is that the molecule has very strong absorption in certain wavelengths, but it must have very low absorption in the other wavelengths if this want to be useful. A lot of the chemicals, they actually have very multiple absorption peaks across the visible wavelengths. For example, if they have there. If that happens, this material is not suitable for us because it's going to strongly absorbing the light. And that's one of the reasons why we tested 20. So in 20 is in the paper, but in reality, we tested 40, 50. That's why in the end, only one or two comes out because of this requirement. We only, in other way of speaking, the absorption peak need to be very clean, uh, so that it don't have additional absorption in the wavelengths interfering with the image.
Am I reading correctly? If
Mm.
your green curve is the absorption peak of the child,
Oh,
like this dye, right? If this is correct,
the the gray curve, the
the gray
dashed curve.
curve, the gray
Yeah.
curve is your dye. Like then I
Yeah.
think most likely you going to
Mhm.
be able to use this dye at a higher like a red or far red, like
Exactly.
it's going to work fine. Or like a very ultraviolet, and maybe kind of can work, but the green spectrum is not going to work, right?
Right. Right. Currently, the tautodyne molecule is not compatible with green fluorescence.
I see, makes sense.
That's why we are using in the fluorescence demonstration. We choose the TT tomato.
Yeah, great. Yeah, if anyone in the audience want to ask question, you can also just send a note, but I will continue ask you one more thing. Like you tried
Mhm.
so it seems that according to your explanation, it's not just like a particular kind of microscopy, like the speckle microscopy. It
Mhm.
can work. It can improve for work for any kind of microscopy. Is that the case?
Exactly. Yes. Yes. Yeah. Yeah. When we a lot of the mesoscopic images are just taken by phone camera. Yeah, a
I
color camera,
see.
CCD. So microscopy, any kind of optical microscopy can be improved. However, there is going to be certain challenges. It's actually similar to the case when we show this epifluorescence. This is like a microscopy imaging of the neurons. You will have a lot of motion artifacts. So I skipped a few slides about data analysis. So it's posing more challenging in terms of how to analyze these data because the animals are still alive.
And it's a bit surprising. You showed one image, one recent study using the mouse on the dorsal side. So that one used is that black 6 mouse? Like, can your method deal with pigmentation as well?
Um,
Maybe I missed that.
so in our case, we mostly use the C57 mice, yeah, black 6 mice. Um, but
Because
the
the
pig, the
belly side is less pigmented, right? But the back is quite pigmented. I'm a bit surprised that you have also tried the pigmentation. It can get
we
rid of.
We cannot. So this is always a challenge in our method is that we reduce scattering. However, for pigmentation, it's absorption. We cannot reduce absorption inside the tissue. I think when they do this experiment, when we do this experiment, if we observe there's certain pigmentation on the mouse body,
To see through.
we won't continue our experiments. We just so
I
we have
see.
to avoid I
I
think
see.
at this moment, we don't have a strategy to clean pigmentation in the mice.
I see. I see. That's cool.
Mm.
Anyone in the audience have questions? Just feel free to interrupt me, and you can unmute yourself.
Can I ask one question?
Sure. Yeah.
Thank you for the nice talk. So my observation of the tissue clearing is having high refractive index is not sufficient to make the tissue transparent. So this is because some chemicals doesn't have the affinity to the tissue. Therefore, the chemicals cannot get into the transparent and cannot homogenize the tissues. So my question is, so what tissues you think this chemical cannot make it clear? And have you ever tested the bone clearing?
This is a great question that we are. So first thing is we are actually trying on bone clearing, but we haven't gone to really characterize that yet. Building a lab takes some time, so but we are actively working on that. A more first part of the question, the affinity of the different molecules. This is a great question that um, if you search, I guess you probably follow some of the tissue clearing literatures. Every year there's a lot of protocols coming out, but reproducing those protocols is challenging. Yeah, so that's because inside the tissue clearing, there's still it's um, a refractive index matching is a general principle that applies. However, there are also other uh, chemical principles behind this, like dissociation of hydrogen bond, like how your chemicals getting infused into the tissue. These are all practical questions. One reason our method is relatively easy is because we are hydrophilic based method. It's just a diffusing, mixing dye with the water. It's a relatively close to tissues component aqueous background. A lot of time if we have difficulty infuse chemicals into the tissue is because these are organic components that are hydrophobic, so it's hard to get into tissue. You probably need to wait days or weeks for the chemicals to diffuse in. While for hydrophilic method, it's relatively faster. Um, that's one of the things I could comment why our method is relatively fast for these kind of tissue applications.
So have you ever compared the ex vivo, so using like a fixed mouse brain, whatever, compare your dyes and with other clearing techniques,
right.
then
That's
the safe
something
speed.
exactly. These are things we are doing now. That's a great question because um, our method we emphasize the safety of the chemicals. That's why we in our first demonstration we emphasize on the live animals, but we also can be used on ex vivo animals. But currently we don't have sufficient enough experimental results to publish a ex vivo paper. So we are actively accumulating more data to test if our method going to be more better or not as good as existing ex vivo method.
Since you are just talking about the safety issue, I
Mhm.
noted that many people might have also noted that the effective dose that you have to apply this
Yeah.
dye is at almost the molar range. That's
Yeah.
really high. And is there any concern about the
Mhm.
adverse effect with using such high concentration
Right.
of the dye? And is there any way like you're making efforts to making dyes that are effective at lower dose?
These are we are actively working on this. So this current dose is relatively high, and for mouse applications, local high dose mouse can recover, but mouse can survive. It doesn't mean it's not toxic. So it will have certain irritation or um, leaving some of the damage on the skin. But for our application, I think it's already a bigger step improvement than all the invasive method that people used in the past.
But how you see is possible to like, are you going to use the same molecule like a backbone as a starting point to make
Oh,
it
I see.
effective at lower dose? Is that possible, or you have to look for other alternative chemicals?
Right. This is something that our group is actually working on. So we hope to build up. This is more like I think the question is more close to a materials question. How do we find molecules absorbs very strongly? Stronger molecules absorbs light. So these are something that we are actually trying to build up a database and utilizing machine learning, artificial intelligence to help us guide what are the potential directions for synthesis. So that we are trying to put together. One reason is because this is a relatively unique angle for looking for tissue clearing chemicals. So we need to build our own database for this. So we hope that we can find molecules that can actually reduce the dose. So another alternative is if something I mentioned in the talk is that if you go to longer and longer wavelengths, uh, the dose you can use we can use different molecules. So currently tyrosine, it kind of absorbs light below 550 nanometer. If your application window is in 700 or 800, we can bring this even further. And using longer wavelengths, actually, uh, using dyes that absorbs at longer wavelengths, this can actually reduce the dosage as well. So that's another strategy to kind of reduce the dosage rate. Yeah.
I see.
Mhm.
A more general comment is that this seems to be a very unconventional way of doing the clearing.
Yeah.
It's a bit surprising because people have tried to adjust the
Mhm.
refractive RI, refractive
Yeah.
index, but this I think I don't know whether it's because I haven't read so much about it. It's like one of few times first time I heard about playing with the absorptions to do it, and it's a bit counterintuitive to our life
Yeah.
experience. How
Mhm.
is it that nobody think about it this way,
Mhm.
or like I don't know, like is it relatable to our living experience or any other phenomenon that you can see this in play, this absorbance making things more clear? Do you see that in real life in other experience?
I actually don't. I think this is a great question, but I don't see that. The simple the most close, I think this is actually some of the reviewers' comments as well. Some of the reviewers gave us some examples in life, and one of the example is, different apples have different colors, like red, green, um, and they noticed that um, different colored apples has different capability in scattering the light. So these different colors of apples actually has scatters light at different wavelengths. That's one of the live example have, but I guess it's still very far away from what we can intuitively understand, right? I think then another part about the tissue clearing community, they are actually getting some of that. They are getting some of them are really excited about our work. Some of them are very conservative about our work. So there are kind of mixed feelings because there in the community, this tested thousands of different chemicals for tissue clearing already, because it's a field like over the past 2 decades. But when they test the chemicals, they have this pre-assumption that these chemicals need to be transparent. So with this pre-assumption, you will never come to what we find. So I think that's the interesting part of physics, why our method is different. We are contributing new thoughts to a well-established field, is that we have some new physical understanding, and we introducing some new design that people excluded in the past.
Thank you. Yao is from Ali Pasha's lab. You want to ask a
thank-
question?
Yeah, sure. Hi, fantastic research. So
Yeah,
our lab right here is doing the mice brain imaging.
Uh-huh.
So actually, one of my colleagues, he actually tried many different concentration of your molecule
Mhm.
to inject them to the mice brain.
Oh,
But
indeed.
most of those mice just die after like 10 minutes or so. I mean, at very low concentration, I believe like the mice can survive. But at that low concentration, like the mice, like the brain tissue doesn't really become very transparent, so you don't really gain too much. So what I'm wondering is like, do you know roughly like what is the mechanism that is killing the mice when you are injecting the molecule to their brain?
Yeah, wonderful question. So these are excellent. You asked this. So one reason we work with skin, skin is a relatively insensitive organ.
Yes.
Different from brain. We did some brain slice. So obviously, if we can clear the brain, this is going to be a significantly big achievement.
Yeah.
However, when I was approaching this question, we prepared some of the brain slices, and we used calcium imaging. We try to see if we add these dye molecules, what will happen. We see that we see random firing patterns. That's why we feel like at this moment, at least for this chemical, we don't think is suitable for brain clearing.
Okay.
You're right. Yeah, this is a great question. And for our brain imaging, we are playing on the skull, and there's a we apply, right. We actually apply on the scalp, so on the skin, and then there's a scalp protecting it, so dye won't use that into the brain, and mice can recover. This is that's very different from injecting into the brain.
Okay. So the reason why I'm asking like the mechanism of killing the mice is like I'm thinking about some other model animals. Let's say Saligans. Let's say in Saligans, like it doesn't have the heart, because we are thinking like it's because like the chemical is actually introducing some whatever reaction to the heart of the mice, then it's killing the mice. But like some other animal like the Saligans, that doesn't really have a heart. So I'm wondering, like, have you know somebody has ever tried this chemical of the Saligans to see like if it's really clearing the Saligans? I know that one is very small, but still like clearing is better for visualization.
Yeah, we haven't. So one of our colleagues, we are trying to work with zebrafish, but I don't know anyone working with
Saligans.
Right.
Okay.
Another, if you are going to do that, one side note is you do not necessarily need to make it. You do not necessarily need to use a very high concentration.
Yeah, sure.
Yeah, introducing this, we are trying to reduce this refractive index gap.
Yeah.
And introducing a little will contribute a little. So
yeah, okay, that should be sufficient.
Right. So you do not. It's not necessary to make it 100% transparent, because it really depends on what application you need. Yeah.
Yeah, makes sense. Yeah, so I might try that out. Thank you.
Thank you. I'm excited.
Thank you. Yeah.
Yeah, I guess the worm usually they don't need further clearing. This is already almost a transparent organism. So in terms of like the potential toxicity you just mentioned, you said that this, when you apply topically, it's reversible and you can wash them away. How long have you kept the mouse after you apply and wash it away? Like you noticed any abnormal thing, any inflammation? What happened to the skin? Like you notice anything when you do long term?
Right. So we actually did this. So when they apply this, so this is some results I didn't have time to show. So we can make it transparent, and through this head, you can actually, skin are still there, but you can visualize the blood vessels on the brain. And then if you wash it away, skin will go back to the original. Still have some yellow tint, but skin will go back to the opaque. So after this, if you wait for it, sometimes mice will form a scar on the region we applied. There's maybe some still some cellular death because of these chemicals, but sometimes this may be related to the massaging as well. So if you massage the skin of the mice quite some time, it will also cause some damage. So currently we are trying to find better ways to deliver this method. That's something we are working on as well. Right.
In your paper, you also showed that there is this gel you would apply, right? It's
Yeah.
you
So
would mix it.
for the difference between when we switch from solution to gel, is for live animal applications. When we want to introducing this molecule into the tissue, we want it to stay on the, for example, we want to apply this on the skin. And then if you just add water or solution onto the skins, usually solution will flow away. But that's why we add usually 0.3 to 0.6% agarose to increase the viscosity of this solution, basically making a soft gel. So we can use a pipette tip to suck the gel and then apply to the region, and then this will stay there for a relatively long time.
Alison is the director of our imaging core. Do you want to ask your question here? Are you here?
I'm here. I think I'm. Hi.
Yes.
I'm so sorry. I missed the beginning of the talk, but I caught the. I was stuck doing something in the facility, but I caught the end. It's super exciting. It's also, by the way, hilarious because the imaging course I run, we've usually added deliberately things from Cheetos to try and make it to demonstrate how you get increased scattering, and that actually is bad for your resolution. So we all love this. But anyway, I'm just thinking away now that just for our scape system. So we've got this newly built system, so we can image things really, really fast. So we don't have the problem of like the gut moving. We can go super fast. But I'm thinking this should be a good way. Have you tried it with escape or oblique illumination system at all, or not yet?
I would say that's a fascinating instrument. I would love to work with, but I don't have the access to that.
You'll have to come and visit.
Yeah, exactly. I think that's one of the, unfortunately, that's one using ultra fast camera is one way to go over the motion. But currently, I don't have the resource to
That's
get
okay. I just wondered. Please say hi to Julia.
Exactly.
I think she used to be one of our users. But
oh, really?
I
Thank
think
you.
so. Yeah.
Oh, thank you. So nice. Yeah.
So I believe earlier Ted asked a question. Maybe you answered half of it. I don't know whether you specifically addressed whether this dye, you see the effect of this dye in clearing hard tissue like the bone. Did you get to the bone issue?
We haven't done too much on this yet. Usually for bone tissue, but from the we read some literature. Usually for bone because it has a lot of salt, calcium phosphate, these kind of salt inside. The refractive index of these salt are very high. Generally, people need to decalcifinate the tissue. We haven't developed this yet. This is something we are working on. So I cannot give a definite answer on how we can make that happen.
Let's see. Anyone else have a question? Please unmute yourself if you. All right, we are about time, but thank you so much for sharing your exciting discovery with us. And
Thank you
I
so
look
much.
forward to catching up with you again sometime later. It's so
Yeah,
nice to
right.
see you. Thank you very much. Good luck with everything.
Yeah, thank you so much. Happy Thanksgiving.
Yeah, bye bye. Thank you.
Bye.
Thank you all.